Assalamualaikum and good afternoon everyone. So today we are going to do our third practical for this semester, which is regarding the topic of sensory system. If you guys still remember our previous topic, on our lessons, sensory transductions, and what is actually the purpose of having senses? So having senses is actually to bring the information about the outside world into the body. So that is actually the purpose of we as a human or organism of having a senses. So about this classification of sensory system, we are based on two, which is the first one is based on stimulus locations. So we have three, if you still remember. The first one we have telereceptors, and the second one we have the exteroreceptors, and the third one we have interoceptor. So this is actually the classification that based on the stimulus location. For the telereceptor, it actually detect the distant stimuli. For example, we have vision, hearing, and for the second classifications that based on stimulus location is the exteroceptor, which is to detect the stimuli on outside of the body. For example, the pressure and temperature. And the third category is the interoceptor, which is to detect the stimuli inside of the body. So this is the materials that we are going to use for today's practical. So this is the dissecting microscope that we are going to use to observe the blowfly, the appendages of blowfly. So this is the adult blowflies, the samples that we are going to use today. And this is the 5 different solutions, that we are going to test on the blowfly. We have the distilled water here. We also have 0.01 mole sucrose, 0.05 mole sucrose, 1 mole sucrose, and the last one we have 5 ppt of seawater. And this is the retort stand, that we are going to use to attach the blowfly on it. So we have the glue. We also have the filter paper. For method, for objective 1, we are going to focus on objective 1 first. So the first method, we are using a plastic bag. So we use the plastic bag and we remove the 5 individuals of blowfly from the insect box. After we capture all the blowflies, then we need to anesthetize the blowflies by freezing at 0 degrees Celsius from 3 to 5 minutes. First solution, distilled water, individual 1. Second individual, third individual, fourth individual, individual 5. Solution on 0.01 mole sucrose. Individual 1, individual 2, individual 3, mole, individual 4, individual 5, we are going to do for the third solution for this test, which is a 0.05 mole of sucrose. First individual, Individu 2. Individu 3. Four. Last, individu 5. Balik. Okay, so we move to our fourth solution, which is 1.0 mole of sucrose. Individu 1. Okay. Individu 2. Individu 3. Individu 4. And last individu, individu 5. Okay, so for the last solution is 5 ppt of seawater. Individu 1. Individu 2. Done. 3.
Lagi basah sikit.
Dah. Individu 4.
Alhamdulillah.
Individu 5. Okay, now we have come to the result sections. So in this result section, you just need to draw a table, and for each individual of blowfly, you just need to tick dash for the negative response and plus for the positive response to each solutions. Okay, so we have 5 solutions in here. We have distilled water. We also have 0.01 mole of sucrose, 0.05 mole of sucrose, and 1 mole of sucrose. And the last one, we have 5 ppt of seawater. Alright, so you just need to compare the result for each replicate, and then you need to discuss it in the discussion session. Okay, so for the objective number 2, so you just need to draw the morphology of the blowfly and label the important characters. Okay. Okay, now we move to the discussion and conclusion sections. So in here, you just write based on your result, what are the expected result that you should get from it, and is that consistent with the practical result or not? So and also you can write what are the characters that play the important roles on cells and etc. And lastly, you just need to answer the questions that I have given to you. So you have 3 questions to be answered. Thank you. See you next time.